We are now live, so good afternoon. My name is Daniel de Oliveira. I'm a professor from Institute of Computing from Federal Fluminense University, and I'm the General Chair of BSB 2020. On behalf of the Organization Committee of the 2020 Brazilian Symposium Bioinformatics, we would like to welcome you to join this year's event. The Brazilian Symposium Bioinformatics is an international conference that covers all aspects of bioinformatics and computational and system biology. BSB is organized by the Special Interest Group in Computational Biology of the Brazilian Computer Society. BSB is a forum for present leading work on the latest fundamental advances in state of art and practice of bioinformatics and related areas. During the event, researchers, students are invited to present their work and discuss their ideas. However, this year, BSB has changed a little bit. For the first time in history, we have an online version of the event. The safety and well-being of our community was our top priority. BSB 2020 could not take place in Roos in August as planned, so the organizing committee of BSB 2020 were continuously monitoring the global COVID-19 pandemic and decided to have a 100% online event. To organize an online event is far from easy and requires the support of many people. I would like to offer my gratitude to SBC, especially to Eli Magario for her support on the organization, FAPERJ for sponsoring the event, Professor Raquel Minardi from Federal University of Minas Gerais for social media support, Professor Sérgio Lifshitz from Pontifício Católico University of Rio de Janeiro for his ideas, and Professors João Setúbal and Valdeir Mendes for their great effort to make an excellent final program to BSB 2020. In particular, we would like to give our thanks to all authors who submitted their papers, to the program committee members and external reviewers that contributed their valuable time and expertise to provide professional reviews working under a very tight schedule. Moreover, we are very grateful to our keynote speakers who have kindly accepted our invitation to give insightful and prospectful talks. Finally, I sincerely hope that BSB 2020 will provide a very good opportunity for you to learn from each other. Welcome to BSB, and I hope you enjoy the event. Now, I would like to introduce Professor José Viterbo, Director of Publication from the Brazilian Computer Society. Viterbo.
Thank you very much, Daniel. Good afternoon. I'm here representing the President and the Board of Directors of the Brazilian Computing Society. And first of all, I would like to congratulate the other participants in this opening ceremony, who are directly involved in the organization of the 13th Brazilian Symposium on Bioinformatics, Daniel Oliveira, Luiz Coada, João Setúbal, and Valdeir Silva. I would like also to congratulate the entire computational biology community that is watching us here on Zoom, or some that are on YouTube also. This year has been very difficult for everyone, and both personally and professionally, I would say. And for the Brazilian Computing Society, it was no different. Like any organization, we had to adapt the work routine of our employees at our headquarters in Porto Alegre. They had to start to fulfill their work activities at home, and it happened so smoothly that many didn't notice the difference in the services provided by our team. But for us, whose the main activity involves holding large face-to-face meetings, there was something more. We had to quickly define and disclose general guidelines for our special interest groups, our comissões especiais, as we say in Portuguese, so that they could be able to plan, adapt, and hold their conferences and workshops online. Again, we did it timely, and eventually less than 5% of our annual events were not able to hold an edition this year. That was a great result for us. On the contrary, some events even grew bigger. In some cases, the number of articles submitted and accepted for presentation was even greater than in previous editions. Not to mention the increase in the number of attendees in the online format. This way, despite all contingencies, we rejoice that the Brazilian computing community remained integrated and productive, offering its contribution to scientific progress and even collaborating the development of tools to support the fight against COVID-19. In this scenario, being here with you to attend a new edition of BSB and to listen to the presentation of the invited speaker, Professor David Roos, is something of great importance for me and for the Brazilian Computing Society. I hope this edition of the event is really successful and that it can reinforce the bonds of collaboration between all people involved, organizers and participants. Thank you very much.
Thank you, Viterbo. Next, I would like to introduce to you Professor Roos Coada, Chair of the Special Interest Group in Computation of the Brazilian Computer Society. Coada. Your microphone is off, Coada.
Sorry. Good afternoon
to
all participants to BSB. I'm very happy to announce that this event is taking place and it's moving forward despite the complicated days we are going through. At the same time, I'm very sorry for not being able to meet you all in person. The event has been happening since 2002 with the name Brazilian Workshop on Bioinformatics. In 2005, it changed its name to current name, Brazilian Symposium on Bioinformatics. For a few years, it didn't occur. In 2018, the CA by Comp was able to continue to organize the BSB event once again yearly. Now, more than ever, we are seeing the importance and the relevance of the research themes investigated and present at the event. On behalf of the Commission Special of the Biologia Computacional, the Special Committee on Computational Biology, I would like to express all my gratitude to Professor Daniel de Oliveira, who has accepted the challenge of organizing the event in this remote form, and with few researchers at his sponsor. I would also like to express my gratitude to Professors John Carlos Setúbal and Valdeir Mendes, who have coordinated the program committee with the due diligence as to include as many works as currently possible. I also thank Professor Sergio Lifetis, who is responsible for bringing us the platform with which we can do the sound and the video transmissions we are currently using for making the event possible. I also give my thanks to all other members of program committee and all reviews. I'm giving my thanks to all volunteers who are kindly supporting us with the required task and event like to this requires. Let me also thanks all research who have submitted their works and trust us with the choice. Finally, I thank every participant that has given us the honor of your presence here with us. It is my hope that you will enjoy it. Please don't hesitate to get touch me if you think there are anything I can to make your experience better. Thank you.
Thank you, Coada. Finally, I would like to introduce you Professors Valdeir Mendes and João Setúbal, TPC chairs of BSB 2020. Valdeir.
Hello, good afternoon. As said, my name is Valdeir. I work as a lecturer in the Federal Institute of Goiás, And this year, I'm part of the Brazilian Symposium on Bioinformatics team, short BSB. As mentioned before, the BSB is an international conference of the Brazilian Computer Society covering bioinformatics, computational and systems biology. And it has 18 years of history. This year, we successfully engaged the Brazilian and foreign community sharing excellent works in diverse areas of bioinformatics. In fact, we had 20 full papers and 5 short papers accepted that will be presented during this event, and that will be published by Springer as a volume of lecture notes in the bioinformatics series. Well, the BSB results from many volunteers' efforts who dedicated their precious time to light these fruitful scientific discussions for the area. And well, there are many institutions and people to thank, but my colleagues already did it accurately. So I do have particular feelings for the BSB because it triggered my choice as a bioinformatician in 2011 edition. For the first time, I watched Professor João Setúbal and Peter Stade, and I confess that at that time I understood very few things, but I felt attracted by the theme and I started to study it. And today I'm here together with Professor Setúbal, Professor Daniel, Coada, helping on the BSB organization. I'm sharing this personal fit because I believe in the power of a strong community to help the individuals achieve their goals in many ways. That's why I always enjoy this event, and I hope you enjoy it too. Thank you very much.
Thank you, Valdir. Professor Setúbal.
Thank you, Daniel. So it is my privilege and honor to introduce you to our first invited speaker, who is David Roos from the University of Pennsylvania in Philadelphia, US. So David is a professor of biology at UPenn. He holds an endowed chair, Otis Kendall Professor of Biology, and he has made numerous contributions to the fields, to various fields, which I have listed here from his website: cell and developmental biology, evolution, genetics, epigenetics, genomics, microbiology, and last but not least, computational biology and bioinformatics. His distinguished scientific career has spanned more than 40 years now. Currently, David is perhaps best known for being the leader and principal investigator of the UPathDB project, the eukaryotic pathogen vector and host informatics resource. Recently, it has changed a little bit its acronym. It has added a V in front of the U, so I'm not sure how to pronounce it exactly. I'm sure that David will tell us how to do it. And I like to note that this started a long time ago, perhaps as the PlasmoDB resource in the 90s, which contained a lot of information about Plasmodium genomics. And this initiative of David has been enormously useful and influential. And not least for people who live in developing countries, such as Brazil, because many of these eukaryotic pathogens that are handled by David's databases and systems are responsible for diseases that are still a problem, a public health problem here in Brazil. So I'd like to welcome David to BSB 2020, and thank you so much for having accepted our invitation. David, the floor is yours.
Thank you very much, João. It's a real privilege and a pleasure to be here with you and your colleagues. It would be more enjoyable if we could be together in Buzios, but I look forward to joining you for some shrimp and beer on the beach at some point in future, and especially to meeting other members of the BSB Society. Just to confirm, I assume that you can see my slide. Is that correct?
Yes, and I can hear you loud and clear.
All right, excellent. Well, as I say, thank you very much for the invitation to present. I feel a little intimidated for a couple of reasons, because like the other introductory speakers, João Setúbal and his former colleague João Amedenes were some of those in highlighting to me the potential of bioinformatics work from their pioneering bioinformatics textbook published, I don't know, many years ago. You accused me of being in science for 40 years, which seemed to me like it's too long, but maybe it's actually accurate. That textbook came out about the time of my first visit to Brazil many years ago. Must have been about almost 30 years ago, and has proved to be quite influential in the work that we do. Now, I should begin with an apology by saying that, pointing out that I'm not actually a bioinformaticist. I'm a biologist, cell biologist, molecular geneticist. I have worked in, I've taken advantage of bioinformatics approaches for many years, and have provide employment to bioinformaticists working in my group and elsewhere. But so this presentation will not focus on the kinds of bioinformatics presentations and algorithmic approaches that I trust will be the focus of most of the meeting. I'd like to spend this opening session to talk about what we all recognize is the most important aspect of what we do, ensuring that the tools that we develop or use are practically useful and get out there to investigators interested in taking advantage of these resources. And I will do so by providing a couple of examples from bioinformatics websites that my group is responsible to, beginning with what João describes as that newly re-acronymized ViewPathDB database, the eukaryotic pathogen vector and host informatics resource, which is designed to be able to capture and provide a wide range of information about genes, but also organisms and various other data types. If my animations are working here properly, there we are. You can see a large number of data types that are recognized, not just including genes, but also organisms and genomic sequences and information that scrolls past the screen much faster than you can see. And I also apologize that today we will be looking solely at screen dumps, because the running the website in the background in conjunction with Zoom's effort to commandeer bandwidth was not very successful in that trial run. This resource, a large bioinformatics resource center for infectious diseases, encompasses a variety of other prior resources, including a fungal database. My group was responsible for integrating a wide range of pathogen informatics data. The Plasmodium genome database for harboring information related to malaria parasites as an indicator of many other parasite database resources that are supported by this resource. And most recently, the V in ViewPathDB vector information relating to insect vectors of disease, whether they are in Brazil or in the US or elsewhere around the world, including all kinds of information you might be interested in exploring, the species distribution, insecticide susceptibility, and so on. In the first part of my talk, I will provide some examples of the ViewPathDB database, highlighting the bioinformatics tools that we seek to bring to the biomedical research community. And I'll close by suggesting some areas that it might be possible to work with other bioinformaticists, such as those who are participating in the BMB meeting. And then I'd like to turn my attention briefly to some more recent resources we've been responsible for developing, dealing with clinical, field, and epidemiological data, the ClinEpiDB project, and an associated microbiome database. There are some references down below, and I'm happy to make these slides available. The first of these is one of many summaries of the ViewPathDB database. The second describes the ClinEpiDB database and a recent application of that. I should have included a reference here for the microbiome DB resource, but you can find that yourself quite easily by typing microbiomedb.org into any web browser. Now, by way of background, as I said, my training is not in bioinformatics. I did work as a programmer many years ago before I got interested in biology, but became captivated by cell biology and evolutionary biology as an undergraduate, and ran a large research laboratory for many years, working on various aspects of parasite cell biology, including the organization and evolution of subcellular organelles that distinguish eukaryotic cells, and particularly pathogens, which allowed us to explore the interaction between these parasites and their host cells. This work sort of culminated in a manifesto describing the field of evolutionary cell biology. And to give you a one-slide example of my former work in evolutionary cell biology, I will provide an example that led to the discovery of the apicomplexan plastid, a chloroplast commandeered by ancestral malaria and related parasites, which is now widely viewed as a promising target for chemotherapeutic intervention. The origin of this work came from our effort to try to identify the mechanism of action of a classic antibacterial antibiotic. In this case, I think this is clindamycin, that is effective against eukaryotic parasites, Toxoplasma, Plasmodium, and a wide range of other pathogens. Now, this is a surprise because what makes antibacterial antibiotics effective is that they kill bacteria without killing eukaryotic organisms such as ourselves. And so we set out to try to identify what the mechanism of action might be, initially suspecting that these drugs, which are well-known protein synthesis inhibitors, would inhibit protein synthesis on the cytoplasmic ribosomes of malaria parasites, but not their human hosts. But we quickly showed that that was not the case. And yet, the activity of a wide variety of structurally unrelated compounds that share in common only their inhibition of transpeptidation on bacterial ribosomes suggested that there must be some other ribosome, which drew our attention to a then mysterious episomal DNA, originally thought to be the mitochondrial genome of these parasites. But when the parasite's true mitochondrial genome was discovered, it left this circular 35 kb circular DNA as a bit of a molecular biological mystery and an orphan without a home. It did provide the only plausible target for these antibiotics. We cloned and sequenced the episomal DNA. Phylogeny told us that phylogenetic analysis of well-conserved genes on that genome told us that the apicomplexa that you can't really see here, but which are highlighted in red, form a distinct clade as we would have expected. That derives from the bacterial world, not betraying an ancestor of alpha proteobacteria that gave rise to mitochondria, but looks for all the world like a cyanobacteria ancestor of chloroplasts. What's more, it looks not just like another independent invasion of a cyanobacterial ancestor to give rise to a plastid in apicomplexan parasites, but it looks for all the world like it derives from the actual chloroplasts of green plants and algae, suggesting that what we think of as the origin of these organisms, distinct from plants and algae, might be incorrect. Or alternatively, that the apicoplast, the apicomplexan plastid, might have arisen by a horizontal transfer in which an ancestral parasite ate or was invaded by a eukaryotic alga and retained the algal plastid. If indeed this two-step endosymbiotic process were to occur by the same topological mechanisms that give rise to the double membrane of a mitochondrion, we would expect to see 4 membranes, and that reminded us of a cell biological mystery known for decades of a distinctive organelle associated with these parasites, surrounded by not 1, not 2, but 4 distinct membranes. And indeed, in situ hybridization allowed us to localize some of the protein products from this episomal DNA to the apicoplast. And we could use cut and paste molecular genetic approaches to define the targeting signals that lead to trafficking into the organelle. Now, this was pretty exciting because it's not every day that you discover a new organelle, especially in a new organelle in an organism that we care about as the source of so much human disease and suffering. But what we really wanted to know was what this organelle did, and its genome gave us no clue, because the 50 some odd genes encoded on this genome encode proteins involved in ribosomal protein synthesis, hence the target of these antibiotics, proteins involved in DNA replication, proteins that might be involved in iron sulfur cluster assembly and other aspects of housekeeping functions, but told us nothing about the metabolic functions which might make this organelle essential and might highlight potential targets for therapeutic intervention. You can imagine a number of molecular genetic or biochemical approaches that we could take to try to characterize the function of this organelle. And I can assure you that we've tried all of them, but the most successful approach by far was to take advantage of this information that you can already see on the slide. The phylogenetic history that betrays a genomic organization related to plants and algae, combined with targeting information, in this case, a 2-step targeting system involving the blue and the green bits indicated here. And by scanning the genome to identify genes that have both of these two targeting signals and a phylogenetic affinity with plants and cyanobacteria, revealed several hundred genes, about 10% of the parasite genome, almost all of which we now know are in fact associated with the apicoplast, and many of which are under active development as therapeutic targets. So this, to me, provided a dramatic example of how integrating in silico analysis with validation at the lab bench can dramatically expedite biomedical research. And that provided our entry into the field. The origin of all of the database resources my group has been responsible for really was a pretty simple script written by me and a postdoc that took all of the then nascent genome sequences and random cDNA sequences, expressed sequence tags or ESTs, blasted them against GenBank, and pulled back pulled back anything for which the best blast hit included the word chloroplast or plant in the title. And that simple-minded Perl script, which took 2 or 3 days to run, gave us back the results of everything that we now know to be true for this organelle, a spectacular validation, in my mind, of bioinformatic approaches. But where we might have wanted to use these tools to ask our questions, genes that have targeting signal A and targeting signal B and a particular phylogenetic history, we rapidly realized that other people might want to use the same tools to ask their own questions. And that gave rise to the Plasmodium Genome Database, which is now more than 20 years old. This is a slide I probably have shown in Brazil previously, that betrays in which over the course of more than a decade, every time we generated large-scale data sets, I tried to come up with a graphic image to represent what those might look like. It's gotten a little bit busy over time, and in fact, I've given up trying to update this. You can see a little bit of the apicoblast genome picking around from the back, along with a little bit of the first genetic map of malaria parasites, but you'll also recognize many other large-scale data types, whether they are transcriptional profiling data from glass slide arrays or photolithographic arrays or RNA-seq experiments, proteomic data indicated here on the upper right, large-scale phenotypic information, in this case for trypanosome parasites, the results of high-throughput screens, in this case on host cells with cDNA overexpression libraries or siRNA knockdown libraries, small molecule inhibitors, structural genomics data, metabolic pathway data in both the pathogen and the host cell, or even clinical data sets that arise, in this case, from studies of malaria isolates in Africa. All of these provide very large-scale data sets, which are the resources that all of us are seeking to mine from the standpoint of exploring biomedical data sets. And I will suggest that there are particular advantages to working on pathogen genomics resources, because to a first approximation, when we're looking at pathogens, we're interested in things that are common, because what our goal is to kill the organism rather than to understand smaller events. And increasingly, we are looking to try to integrate information that comes from many different sources, from many different investigators. All of these papers describe genome scale phenotypic characterization of various different pathogens, none of them coming from my research laboratory, but where we are responsible for integrating that data into the Plasmodium Genome Database, initially released in 2000. And now the ViewPathDB Bioinformatics Resource Center, a project that supports more than 40 staff, includes many hundreds of unique data sets of a wide range of types. I won't read through this, you can read it yourself, and I'm happy to make these slides available for anyone who would like to take a look at that. Thousands of different data sets, thousands of metadata terms that we're responsible for harmonizing in ways that allow users to integrate these diverse data types. Genome sequences, yes, but also annotated information that comes from the community, variation data from population studies, metagenomic and metatranscriptomic information, proteomics, transcriptomics, metabolomics, geolocation data, and so on. There are many ways that we seek to integrate this information. Here, a JBrowse view of some of the information that our users might want to use to identify, for example, the alternatively processed transcripts. In this case, in the Toxoplasma genome, a gene cloned in my laboratory 20 years ago that was the first identified case of alternative splicing in Toxoplasma. We can use this information to overlay information from RNA-seq, deep RNA-seq data sets, single-cell RNA-seq data sets that we couldn't have dreamed of 20 years ago, transcription factor binding sites, comparative genomics data, population genetic information, all with an eye towards trying to understand and elucidate what the functions and proper annotations of these genes are. There are a myriad of interesting questions that I'd be happy to discuss with people afterwards. Sadly, not over a caipirinha this evening, but certainly during the question session, or for any of you who would like to contact me afterwards. Our goal of integrating information comes not just from browser views, as indicated on the left, but also the desire to integrate information, in this case in the center, from a wide range of transcriptomic analyses. This is a summary tool that we've developed to try to allow users to look at an individual gene across all of the experiments that have been published for this particular gene. And we are interested in exploring machine learning-based approaches to highlight what might be interesting for this. You would imagine that for an investigator who might be interested in this particular gene, NCU 0007, whatever that may be, we would like to be able to highlight to users that this gene is interestingly regulated in experiment X or under condition Y. We'd like to be able to link that to transcriptomic data here, metabolic pathway maps, which are painted with phylogenetic profiling data, painted with transcriptomic profiling data, inferred information across species. Here are results from the OrthoMCL ortholog identification algorithm developed in my laboratory years ago, and also with phenotypic information, whether this comes from large-scale genomic tagging experiments or for, in this case, a genome-wide CRISPR screen to knock down that information. From the standpoint of participants in the BMB project, from the BMB Society, there's a wealth of information here that is available today for you to download and analyze. The inadequacy of some of these visualizations highlight potential for you and others to develop tools that we, for certain, would like to be able to take advantage of, and maybe we can talk about that a little bit later on. Some of the tools we support include browsers, such as the browsers that you've seen as products of the GMOD consortium, an annotation platform we're rolling out using the Apollo platform, which has also been developed as part of the GMOD project, the ability for users to download specific information that they're interested in, or to upload information that they may have, for example, a proteome of a new organism that's been sequenced to define putative orthologs that can be used for in silico annotation. Web services that provide API access for programmatic access, and increasingly tools that allow users to analyze this data for themselves, such as the ability to paint metabolic pathway maps with phylogenetic profile data and transcriptomic data and metabolite data, as I've shown you previously. The ability to map information that is linked to geostratigraphic information using semantic zooming to zoom in and look at higher levels of detail, in this case on mosquito species distributed through the state of Iowa in the US. And these semi-graphical browsers that allow users to integrate information and identify features of interest, in this case in the vector-based database responsible for housing data on mosquito species, identifying secreted regulatory factors that clearly show some enrichment in Go terms. Let me just take you on a brief walk through some of the some similar sorts of queries. Some of this quite old. In this case, an in silico analysis that was carried out by a very talented PhD student in my lab, Lucia Peixoto, who now has her own laboratory at Washington State University. Lucia was interested in asking not about the kinases that we know are likely to regulate signaling function inside parasites, in this case the toxoplasma parasites she was looking at, but asked for those eukaryotic protein kinases that are also predicted to be secreted. Something of a heresy, because we normally think of these kinases as playing a role in intracellular functions. But for an intracellular parasite, secreted kinases might play a role in regulating host cell function. She identified candidate secreted kinases that showed evidence of diversifying selection by virtue of analysis of population genomic data sets that came from others, and expression timing that was done from others as well. And the net result of her study was to identify a series of proteins known as rope tree kinases based on their localization in the distinctive secretory organelles of these parasites. You'll note that nowhere in our search here did we specifically look for secreted kinases, and this gave rise to the successful PhD thesis from Lucia. But because this analysis has been carried out entirely in silico, you can imagine that we can easily transform this search to look not for kinases, but for phosphatases, revealing other candidate genes which have served as the basis for PhD theses in other laboratories around the world interested in exploring this further. Here's another example that occurred to me sitting in the back of a room in a parasitology seminar where I heard a presentation by Peter Myler at the University of Washington, who had identified the first localization by ChIP-seq analysis of an unusual base, DNA base, modified DNA base that had been discovered and characterized by investigators in the Netherlands, and identified this base J as being associated with the termination signals, transcript regions of the very long transcriptomic, the very long polycistronic transcripts in kinetoplastid parasites, such as Trypanosoma cruzi or Trypanosoma brucei or Leishmania. In this case, Peter's studies were done for experimental reasons in Leishmania, and you'll notice that these peaks arise solely at areas of convergent strand switches, and the hypothesis that they might serve as terminators to prevent transcriptional read-through that would inactivate genes downstream. And it occurred to me that if indeed that is the case, we could conduct an in silico experiment because you would predict that essential genes, if it's so important to prevent read-through, essential genes might be preferentially excluded from convergent but not divergent transcript regions in the genome. So I ran a small query here to ask about trypanosome proteins, excluding hypothetical proteins that hadn't been well characterized there, but which we know from the first genome-wide essentiality study conducted by David Horn's lab, now at the University of Dundee in Scotland, were always essential. And now I could ask where those genes lie by forming a genomic collocation query to ask for these proteins that these essential proteins that overlap the downstream region, giving rise to the discovery that very few essential proteins are located close to convergent transcript regions, and simply by changing the Boolean here to ask for the localization of non-essential genes, we find a much greater number that are located in that region. This is not just something that I can do sitting in the back of a seminar, or the student working in my lab can do, but is increasingly a project that others are interested in exploring. This is one of the first successful stories, a project carried out by Isabella Ochola working in Kenya, Kevin Tete then working in the Gambia, and their supervisors working in Kenya and in London were able to formulate a query looking for candidate vaccine antigens in malaria parasites based on criteria that they define, not I, based on their immunoreactivity, their evidence for selection, their localization of expression. And that gave rise in this particular query to a list of about 20 some odd genes, including as some appropriate controls, identifying both of the leading candidates for bloodstream therapeutic targets for vaccines, some of which are in clinical trials now, a number of other genes which have previously been identified as candidate antigens of interest for malaria therapeutics, but also some conserved hypothetical proteins that had not yet previously been identified. And clearly, if you are interested in vaccine development, while no one would argue that any gene on this list will prove to be the long sought effective therapeutic target for malaria vaccines, surely those highlighted in green, which we can identify in a few minutes at the computer, would be of interest to explore. Now, increasingly, we find that rather than coming to database resources to look at large-scale data sets developed by heavily resourced laboratories and genome sequencing centers, increasingly, all biological laboratories are able to generate and analyze their own data, and we can help to facilitate that analysis by making sure that the tools and pipelines that we've developed for the ViewPathDB database are also accessible, in this case, by developing a cloud-based Galaxy platform that allows the many thousands of end users who use these resources to take their own RNA-seq data, point set up an account on our Globus-based cloud-based resource to run a Galaxy pipeline, manipulate that as appropriate, and return the results. In this case, for example, the mapping of RNA-seq data to a large protein in, I think this is probably, no, this is, looks like this is in Toxoplasma parasites. But most importantly, in addition to being able to analyze this themselves using our validated pipelines without having to search for the reference genome that has already been uploaded to the cloud, they can bring back their results to interrogate it in the context of all other available data that is available in ViewPathDB datasets. For example, isolating genes that are differentially expressed in this experiment and also in others. This will become increasingly important as we seek to provide integrated access of information that comes not just from pathogens, but also from hosts. I'm about halfway through, or a little more than halfway through the time that we have available for the presentation today. So I think I will skip over some of this and simply highlight the fact that these tools and resources are only useful if they are actually used by individuals in the broader research community, and we spend a great deal of time and effort trying to assess what the actual access looks like. ViewPathDB resources are currently accessed by about 50,000 unique users per month, and the analysis of users' own data on, for example, our Galaxy, our cloud-based Galaxy platform is accessed by about 2,000 unique users per month. The Apollo annotation platform has only been available for vector-based users, but will be rolled out over the coming months to other taxa as well. And so we look forward to raising these numbers. This includes use by researchers all over the world with a distribution that is not surprising. Heavy usage of the trypanosome database in Brazil, for example, less heavy usage of the Plasmodium database, some interest in other pathogens as well. For those who are interested in the distribution, I'm happy to describe this to you later. Depending on the month, Brazil is typically second only to the UK, sometimes first in its usage of the trypanosome database. And these resources have been cited more than 20,000 times in the scientific literature to date. So I will come back to this at the end to highlight the fact that there are tremendous needs for improved algorithms for data analysis, tools for analyzing that data, tools for visualizing that data, and making this information available publicly. We are consumers of many such tools and would be happy to discuss with any of you who have interesting tools that you think might be worthwhile to us. And from our standpoint, we are also happy to make those tools that you and others develop publicly available in ways that see the usage that they deserve. But I'd like to take the remainder of my time today to talk not about the genomic data sets, which we all recognize have revolutionized biological and medical research over the past generation. No molecular biologist, no immunologist, no cell biologist, no geneticist can afford to be unaware of genomic data sets and informatic approaches. I'd like to take the remainder of my time to highlight some newer resources that even those of you who are familiar with the ViewPathDB family of databases may not be aware of. Some years ago, we were approached by researchers working on a variety of parasites, particularly malaria parasites, pointing out that there were large-scale data sets that were available for malaria, for example, or for many of the diseases that we work on that have no direct link to the genomes that we were trying to map them to, that were instead tied to clinical and field epidemiological studies. This image here represents the kinds of data forms that many of you have dealt with. Certainly Brazil has been a leader in providing high quality epidemiological data sets. Brazil, in fact, was involved in the case report forms described here from the global enteric multilocus study looking at diarrheal disease. Here a study in Africa on respiratory infection. And here another study on enteric disease from a large-scale longitudinal study that I'll say a bit more in the future. And epidemiologists are used to these kinds of problems, the difficulty gaining access to the data to begin with, trying to load that into platforms that are not really suited for dealing with studies like the NALED study that includes millions of data points. So our goal in what has now come to be called the ClinEpiDB project is to provide a user-friendly access for enabling users, whether those are the investigators themselves or their colleagues, in-country public health, in-country public health officials, researchers interested in accessing data for doing the kind of informatics analysis that many in the BRB meeting are interested in, or any other kinds of end users or biologists to be able to analyze that data. Our goal is nothing short of the revolution that we have witnessed through the work of many people, ourselves included, in transforming biomedical research on laboratory studies to see the same sort of revolution in clinical and epidemiological studies, whereby investigators can quickly and easily visualize the data, looking at the analyses that they might want to conduct. So let me take you on a brief walk through clinepidb.org. Although, as with the ViewPathDB database I described, you feel a little bit silly trying to present in platform view what is really a dynamic resource that you are welcome to explore yourself, to search studies or the organisms or any of these things that you're interested in exploring, exploring on your own, exploring the studies, exploring sample searches. When you come to ClinEpiDB, you'll see a wide range of large-scale studies that are available. Other studies that are grayed out here that are in process. Many of these have now come into the public domain with the most recent release, as well as the ability to explore particular sample searches and visualizations, which we hope to improve dramatically over the coming months, perhaps with help from some of you. Now, each of these studies are large studies. The PRISM study that we're looking at here is a longitudinal cohort study that followed over many years the acquisition of functional resistance to malaria at various sites in Uganda, characterized by different entomological inoculation rate, that is, different exposure to parasite-infected mosquitoes. We might want to ask questions of the patients themselves, their clinical visits, the households they live in, or the insect vectors of disease. And for each of these large studies, we provide a study page that allows the investigators to highlight key publications, information on the design of the study, highlighting the 3 different regions where the study was carried out, a flowchart for sample analysis, and contact information for the investigators that are involved. Diving into this, we provide a user-friendly platform for filtering down the data to identify data of interest. And again, if we were doing this live, I'd like to be able to walk through this with you. Here, we're just looking at screenshots, but you should be able to emulate this yourself if you're interested. I have filtered my information on one particular site with a high level of a high malaria burden, looking only at children, And we can see that of the 48,000 clinical samples that are represented in this study over a 5-year period of time, there are 3,000 cases of malaria. By looking just at the visit date, I can see a graph of all of the visits as well as those that satisfy my search criteria for clinical cases of malaria. And immediately this highlights to me some things that I don't understand about the study itself. Clearly, these visits are not uniformly distributed. Something changed here and something changed here. And if I were to go back to that study page, I would see that early in the study, we saw the study was carried out with monthly visits only. Subsequently, more regular visits by a subset of the population, followed by a gradual decline in the number of studies that were looked at. So the denominator in this study is changing over time. But despite that, we can see a very high incidence of malaria, thousands of cases in the early part of the study, but something seems to have changed here, much less malaria. And what's changed was the distribution of interventions, including insecticide treated bed nets and widespread insecticide spraying campaigns. We see not much impact of this distribution of bed nets, but a dramatic impact of carbamate spraying followed by organophosphates later on. The kind of information we want to highlight to users that is brought that may explain these phenomena. Now, that question allowed us to look at cases that were positive for malaria, but by clicking on this, the user can rapidly switch over to looking not at symptomatic malaria, but asymptomatic infection, either by virtue of a blood smear or a molecular diagnostic technique. And when we look at that, we see that in contrast to the uniform distribution of malaria observed earlier, we see a distribution of asymptomatic parasitemia, which correlates early on in the study with their clinical visits, right? Not uniformly distributed. And that's a consequence of this being asymptomatic infection. These kids weren't sick, so they were only diagnosed as having parasites in their blood when they came into the clinic. Consistent with this here, but now we see only a mild diminution in asymptomatic infection with spraying campaigns, suggesting that there's carriage of a large number of asymptomatic parasites in the blood of this population. This particular study has been continued to the present day, and the most recent release of this data has just become public with the release of the last database. Now, what we can see is that that in the more recent study, the PRISM-2 cohort, following on from the previous study, very, very little malaria, symptomatic malaria, a few cases that were positive by blood smear. But when we turn to molecular diagnostic approaches, we see a vast reservoir of parasites persisting in the population, something of critical importance if you're a public health official interested in appropriate interventions. As with our genomic resources, we can dive in to take a look at the individual participants, follow the course of their infection over time, and support complex queries of the form that you've seen for genomic data sets. This is an example I particularly like because this was a query that was formulated by a data manager in Nagongera in the far eastern part of Uganda, close to the Kenyan border, who was interested in knowing how the kinds of houses that people live in, epidemiological characteristics associated with an epidemic might affect the incidence of malaria, and in particular, the age at which children in those households were first diagnosed with asymptomatic parasitemia. Early in life, you'll never see asymptomatic parasitemia. Any child that has malaria in their blood will be sick. But as children develop functional immunity in these hyperendemic areas, we see a gradual acquisition of functional resistance in in these areas of high malaria endemicity in eastern eastern Uganda, as we also see in areas of Amazonia in Brazil. And here, here, this investigator formulated this query by asking about the kinds of houses people looked in at the age of the first diagnosis of asymptomatic parasitemia. And for those who live in more modern housing with a tin roof or a tile roof as opposed to a thatched roof, I think you can see a very dramatic difference in the age at which they're exposed. That allows them to analyze their results, configuring an app written in our Shiny to look at the age distribution in this area, and we can see a shift from thatched roof houses to metal roofed houses over time. The ability to conduct these kinds of queries depends on extensive semantic harmonization, and let me maybe, in the interest of time, skip over the semantic definitions just to direct your attention to a publication describing how ontologies are defined. Perhaps the most important part of the ClinEpidB project, the first thing that we did upon obtaining real funding for this project, in this case from the Bill and Melinda Gates Foundation, was to build an ontology team whose job it is to make sure that, for example, when we talk about modern housing in Uganda and in Amazonia, we're talking about the same thing. When we talk about a fever in Thailand or in Colombia, we're talking about the same thing. We've now defined thousands of ontologically defined terms, which allow semantically defined terms, which allow us to interrogate across specific data sets. And I'll give you a couple of examples of that in closing. So we've looked at the ClinEpidB database before. Now let's turn our attention to the MALI-D study, a longitudinal cohort study that explored a vast amount of information in 9 sites around the world. Again, you can look at the study page yourself to find some information on this, including requesting access to the data, which is now publicly available upon submission of a data request form. Just as you've seen for the PRISM malaria study, in this particular query, I've decided to contrast sites in South Asia and Bangladesh, India, Nepal, and Pakistan with those in Africa or South America, Tanzania, South Africa, Peru, and Brazil. And what you'll notice is that that those households in South Asia were disproportionately had piped water rather than relying on a public tap or an unprotected well or a river for water. And yet, when we turn to look at enteric disease, among the many different criteria that are scrolling by on the screen to the left, we see that, let's see, sorry. When we look at total diarrheal cases, I guess I've left out a slide to show that. What you would have seen is that despite having disproportionately high improved sources of water, they also have a disproportionately high abundance of diarrhea. This slide here illustrates a dramatic, a remarkable aspect of this data set. This is a longitudinal cohort study with daily record collection over the first 2 years of life for 2,000 individuals. Those of you who've been involved in clinical studies can imagine the difficulty of capturing daily information over 2 years of life. And you'll note that for the vast majority of these children, they have 730 daily records, a really rich data set. If we turn, I see, here we are, to look at the cases of diarrhea, you can see that despite the fact that their water supplies were relatively good, South Asian children had a very high incidence of diarrhea. And we can look at the source of that diarrhea by bacteriological analysis, in this case, looking at infection with Campylobacter by traditional bacteriological approaches as compared with molecular diagnostic approaches. where we can examine the PCR results over the cycle times for this study. We can formulate questions in much the same way we did for genomic data earlier on, by asking in this particular case for children, darkening in this area, with a heavy Campylobacter infection based on a bacteriological or PCR assay, who then subsequently were observed to falter in their growth maturation. So I'm asking for children with a heavy Campylobacter infection with a normal height, in this case, height to age, z-score height for their age. And now asking for those that over the subsequent couple of months, I'm going to modify my query to ask over the subsequent 3 months for children that fell off the weight curve. And you'll notice here a very dramatic decline here beyond the two standard deviations from the WHO norms for growth. These are very sick children, and we can then use those to download information about these individual children. All of this aggregate information that we have run is available without access restriction, and access to the underlying data requires approval by the site investigators, which can be granted through the ClinEpi website. Simply requires you to describe what you want to do with the data, how you will analyze it, and you can see as well what other investigators have chosen to explore. Let me give you, in closing, one final example. In this case, looking at another enteric study, the global enteric multi-locus study, a case control study, seeking to ask what it is that children die of when with severe, with moderate to severe diarrhea in childhood. And once again, we can ask at particular sites. In this case, I'm asking for Bangladesh about the incidence of individual infections. Here, looking at Shigella. Some of these children have a very high burden of Shigella, and I am going to compare cases versus controls to ask about the incidence of infection and the age distribution. Not surprisingly, I find that Shigella is most common in young children. These are, it's like in the first year of children, approximately 10 months up to 30 months in age. Rotavirus is observed in even younger children, from 16 months to 18 months, whereas a protozoan parasite like Giardia is observed over a much broader distribution, up to 5 years old, as examined in this study. We can use our Shiny apps to assess contingency tables. In this case, for example, I can look for the impact of norovirus in cases versus controls and find a very little change in risk factor for severe diarrhea, in contrast to Shigella, which shows a high burden of risk all over the world, low burden in Campylobacter infection, higher burden in Cryptosporidium, a protozoan parasite, and remarkably, the observation that Giardia infection seems to be protective for diarrhea. It is more commonly observed in controls than it is in cases, and we can look at that across all of these sites worldwide. What might explain this surprising finding? One obvious possibility might be aspects of the intestinal microbiome, and fortunately, stool samples were collected from these patients and subjected to microbiome analysis, which has been deposited in Microbiome DB, a parallel database using the same underlying infrastructure that we have developed for the ClinEpi DB project, and which I encourage you to take a look at on your own. In this case, we provide data from a large number of studies, including, for example, this GEMS case control study, and this leaves me with the final example of cross-platform integration that I'd like to highlight. So what are we looking at here? In the epidemiology database, ClinEpi DB, we have extensive, well-characterized, ontologically defined metadata associated with this study, allowing us to define exactly the patients that we are interested in. Those, for example, which have severe diarrhea, use a particular water source, live in a certain kind of housing, have socioeconomic characteristics that we might be interested in exploring associated with diarrhea. That level of detail is not available in the microbiome database, but the microbiome database allows us to explore the kinds of parameters we would be interested in for any microbiome study, alpha and beta diversity, for example. So here's what we're looking for. We would like to be able to use the epidemiology database to ask specifically about GEMS cases versus controls, and the microbiome database to look at the distribution, the beta diversity of species in that study. And we can integrate this information to configure a graphical representation, again here done in Shiny, to reveal some interesting insights, in particular, that the Giardia positive cases are particularly rich in Prevotella and depleted of the gamma proteobacteria, which include many of the most pathogenic bacteria, Shigella, for example, E. coli, for example. Whether this is causative or correlative, we don't know, right? One might imagine that Giardia changes the microbiome in a way that discourages infection with pathogenic bacteria, or conversely, it may be that pathogenic bacteria discourage infection with Giardia, or perhaps most likely of all, pre-existing intestinal ecology that supports pathogenic bacterial growth might be disproportionately subject to infection with Giardia. Regardless, you can imagine how in silico experimentation here could drive the next set of experiments quite readily. So let me conclude with just a couple of slides. And as I'd indicated to Joao earlier, the discovery that I couldn't run some of these presentations live via Zoom meant that I only had a few minutes to pull together a couple of quick summaries. I'd like to encourage all of you to explore the ViewPathDB and ClinEpiDB databases that I've talked about, the microbiome database as well. And we would love to hear from you what works, what doesn't, for you as bioinformaticists seeking data, for you as tool developers looking for applications for the tools that you develop, or for you as interested individuals, citizens, and scientists interested in what works and what doesn't. Because the key point that I want to convey is that without the ability to communicate the results of our studies effectively through publication, through online resources, or whatever, there's no point in doing the work that we do. And there are many opportunities for visualizations. We are, for example, currently implementing approaches for analyzing and representing the large-scale data sets that emerge from single-cell RNA-seq studies. And I know that from the abstracts that some of you are interested in this as well, something we'd be happy to talk about. We're certainly interested in implementing new analysis tools that you or others develop. We, for example, are exploring AI-based approaches for summarizing data. For example, suggesting that an investigator who is interested in certain genes might be interested in others, or certain patients might be interested in other studies, and machine learning based approaches for integrating results from distinct studies. Increasingly, we are called upon to provide the tool, to make the tools that we use for the analysis of data loaded into our databases accessible to individual researchers, which we have done by the cloud-based Galaxy pipelines I've described, and by containerizing tools which dramatically lowers the cost for our providing tools to our various clients. And we have an extensive team responsible for various outreach activities. Finally, I'd like to encourage all of you who have been thinking predominantly in the area of genome informatics to think about the epidemiological frontier, something that's very much in all of our minds as we try to adapt to this world characterized by widespread COVID epidemics. Brazil, like the US, is suffering heavily in this front, and we need to help bring the big data revolution to bear on public health problems, and particularly the challenges of cross-silo data integration. I'll close by thanking the people involved in this project. It's a pretty large group, and I would like to point out, interestingly, that as this project has been running for more than 2 decades now, and has come to provide support for staff. This is provided support not just for students and postdocs and independent faculty, but also a whole variety of alternative scientific careers for PhD level biologists and bioinformaticists in all of the areas that are described here. All of those shown in this picture here are staff. Very few of them are faculty. And it's an exciting new world for bioinformaticists with many career opportunities that I'd be happy to discuss if time permits. So with that, I see that I am out of time. So let me thank you very much for your attention. Turn the stage back over to João and the other organizers, and I'm happy to take any questions if time permits.
Excellent, David. Thank you so much for a very nice and stimulating talk. We will now have time for some questions. Questions should be typed in at the chat area. Let's see if there are a few questions here. Well, we do have the first question. Sergio Lifshitz, who's part of the BSB steering committee, asks the following question. Could you please explain further those opportunities involving galaxy pipelines, effective communication issues?
Sure. So I'm happy to talk about that. I trust that most of the members of BMB are familiar with the Galaxy platform. We're big and enthusiastic supporters of the Galaxy Galaxy initiative, and by its very nature, anything that's available in the Galaxy toolshed is available to any of our users. It has taken some engineering on our part to make this accessible to users via a cloud-based interface, and with hopefully a less steep learning curve for non-bioinformaticists. So we've been gratified to see that, as I showed you, on the order of 2,000 users per month of our genomics resources come to ViewPathDB to analyze their own data using the pipelines that we provide for RNA-seq analysis, for SNP calling, for ortholog identification, and for other functionality. We would be happy to make available to users the tools that you may develop in Galaxy, and there are several advantages to users to work through the ViewPathDB interface. First of all, it's free for them. It provides a cloud-based infrastructure for access. Secondly, all of the information that they need for analyzing their data, for example, reference genomes and default parameters, are defined as part of this process. Thirdly, it allows them to bring their results back for analysis and integration with the other resources. And perhaps the greatest possible benefit, which we have not yet capitalized on, is the fact that having already analyzed their data via our validated pipelines, it's already passed our QC. So while we don't know what data people have been analyzing, we ought to be able to give them a little checkbox where they can say, this data looks pretty good to me. Would anyone else like to use it? We can make that data available. Or perhaps a checkbox, an alternative checkbox that says, we're not ready to release this data yet, but ping me again in 6 months' time. And because it's already past QC, it dramatically reduces the time that's required for our data loaders to analyze that. So for BMB investigators, perhaps Sergio or others who may be developing Galaxy pipelines, if there are tools that you have built that you think might be useful to ViewPathDB users, we'd love to hear from you. We'd be happy to work with you on making those tools available to our fairly large client audience. And we are in the process of building similar tools for the microbiome DB and ClinEpiDB database. Joao, you're muted.
Thank you, David. We have another question from Ronnie Alves, who's also from our steering committee. I was wondering about data governance and metadata. I believe these are key for big data revolution. Do you have any tips and tricks on handling those metadata?
Yeah, absolutely. Absolutely. Key question. And I have a couple of comments to make about this. Governance is indeed an immense challenge, and metadata standards are also key. And here's where I have a couple of maybe controversial comments to make, because our approach to handling some of this has been a little bit different than what I think the standard approach in data access has been. As a bioinformaticist, I have suffered through the years of efforts to try to define metadata standards. My lab was one of the first to sign on to pilot application of some of the Miami standards for microarray analysis that some of you may remember. And students I well remember that students and postdocs in my lab worked with my colleague Chris Stockert and others involved in the Miami consortium to define in excruciating detail all of the information that we as biologists typically tend to ignore, even though we know it could be important for understanding a study. And that took weeks or maybe even months. And then when the next study came around, it took weeks or months yet again, and people gave up. So I have pretty strong views about the importance of metadata, but also the need to be able to define standards that users will actually use. And that really requires a rigorous application of appropriately defined ontological framework for these terms. From my standpoint, the greatest benefit of the ClinEpiDB project has been that it has allowed us to hire a professional ontology team, because important as metadata is for lab-based informatics studies, it is critical for clinical studies. The example I always give, for example, is that my laboratory did some of the first transcriptional profiling studies on Toxoplasma parasites in vitro and in vivo. And we didn't really pay too much attention to our studies, and we wanted to compare that with similar studies that were generated by colleagues in Belo Horizonte or in Stanford or in Switzerland. And yet we didn't pay too much attention to what the culture media was we used, what the cells we grew it in, what the mice was that we infected, what the sex of the mice was that we infected, even though it could easily be that parasite transcriptional profiling would be different in a male than a female. Contrast that with your experience if you go into into your physician for an annual checkup. If I go into my physician for an annual checkup and the doctor that I see tells me that it's time for my mammogram, right, I will be looking for a new physician immediately. Right? If that physician hasn't recognized the differences, the importance that gender might play. So I know that no one in this audience is unaware of the importance of metadata. But by the same token, our approach has been not to try to define all of the terms that we need in advance, because we will run the risk that we faced with my student trying to define the criteria for microarray metadata. What we work on is to try to capture the data up front and build trust with the study investigators so that the call for increased definition of metadata comes from the investigators themselves rather than something that a bunch of nerdy bioinformaticists are trying to impose on them. And that has proved to be far more successful. This kind of pull mechanism has proved to be far more successful than a push mechanism. Sorry to ramble on here, but let me just give you an example of that. Our first work with metadata probably related to the capturing of genome annotation from the user community. So we implemented what we called a user comments form that was a free text field that allowed any biological investigator to type in whatever information they knew about a gene. I first reported this at a genome informatics meeting in Las Vegas, I think. And it was maybe the most unpleasant presentation I had ever given, because I was completely raked over the coals by the annotation and curation community, who said, if you allow end users to annotate genes, you're going to get nothing but crap. And this is a disastrous idea. I, on the other hand, had this starry-eyed view that we were going to bring the power of Wikipedia to the analysis of genes. And it turns out that both of us were completely wrong. With a decade's worth of experience, we now have community annotation for tens of thousands of genes. We have almost no garbage. But the utility of that information is dramatically limited by the lack of appropriate metadata standards, as you might imagine. But what we have seen is that our users say, I've just annotated this gene to say that it is turned on under the following conditions. I'd like to be able to find other genes that are turned on under the same conditions. And so they recognize the need for metadata standards, which we have increasingly ramped up. And the opportunities, I think, are immense for this, for our epidemiological resources. So, as I said, sorry for the lengthy comment. I hope that those are informative for you, Ronnie. I haven't actually addressed the issue of governance, but governance is a very closely related issue, and I will give you one example on this from our epidemiological resources. When we embarked on this project, we were warned that there was no point in providing a user-friendly access to clinical and epidemiological resources, because no one would ever agree to let those data go public. We were warned of this by, for example, the principal investigator on the PRISM project that I showed you in Uganda, Moses Kamya. But we embarked on this nevertheless just as a pilot to see if we could do something useful. And something very interesting happened, which was exactly parallel to our experience with genomic data. I will maintain that the users that all of you in this audience are familiar with, who are reluctant to let their data go public and are reluctant to do so for fear of being scooped on their own data, I will maintain that even those who are most protective of the data for fear of being scooped are actually protective of the data for fear of being embarrassed. Right? None of us wants to put data out there that are crap, that just waste people's time. None of us wants to put data out there that will lead somebody else to say, you missed this. You know, how idiotic is that? But the problem with large-scale data sets is that it's so difficult to wrap your head around it. So if we can provide easy, user-friendly tools that allow people to explore their data, they will be the first to recognize how valuable they are publicly, how beneficial it will be to the user community in an altruistic sense, and how much credit they will get by making that data publicly available. And the PRISM project is a great example of that. This was a 5-year, $10 million study for 50 investigators. And all of that data, every bit of it, is currently available publicly at the request of the data providers. So our experience has been that if you need to try to define governance in advance, you will never do it. You will never get to go. And therefore we have a very simple data release policy. We never ever release any data until the data providers agree that it is properly represented and ready to go live. And by not requiring legal sign-off from the outset, we can get to work to figure out if we can do something useful. And when we can, then the data providers have an incentive to work with us to figure out whatever any remaining problems may be. Sorry to be rambling. This isn't a very articulate explanation, but I hope that that makes clear. I do see a note from Ronnie Alves saying he agrees with that comment.
Thanks, David. It sounds like you can read the questions for yourself. We have another question from Valdeir.
Yeah, I can. I see a question from Valdeir asking more practically whether the ViewPathDB family of databases have full computational access. Yes, everything that we provide is fully web services enabled. The APIs for accessing it are not necessarily entirely clear, but the easiest way to access things is to click on the Contact Us link at the top of any page, and that enters into a tracking system so that folks on our outreach staff will get back to you about what you need access to. The same is true from the epidemiology databases. There, because of the complexity of the projects and the people who need sign off from them, it may take some time to gain access to it, but everything we do is web services accessible.
Great. I have a question of my own, David. It's not a technical question. So you've been working on this field for at least 20 years. And I believe that it was back in 2004 or 2003 that NIAID decided to fund these bioinformatics resource centers. And I think at the time, one of the incentives for doing that was the SARS epidemic in China. And the head of NIAID at that time was Tony Fauci, and he's still there. But he became a much more famous person now than he was 16 years ago. So the question is, you've been working on this for 20 years, and that's infectious diseases. Infectious diseases became a very prominent subject this year. Of course, this has had an enormous impact on all of our lives. But I wonder, for you, who's been working funded by NIAID, what kinds of impacts that you have seen, even though you don't work directly with COVID-19. I would imagine that NIAID has changed because of the pandemic. How does that trickle down to you?
So certainly I view the impact of this pandemic. I mean, so a couple of things I should comment on. Certainly, you're correct that the largest single source of support for the ViewPathDB database has come through the Bioinformatics Center contract which you know very well because you were involved in the first round of BRC funding. That project was funded as a contract rather than a grant, which at the time I thought was ridiculous. I'm a research scientist and I lived in grant world. And in retrospect, I have come to recognize that that was a very insightful change of heart. It's something that I would like to urge on FAPESP and other funding agencies in Brazil to think about service resources, the need to support service resources as resources with an obligation to the community. For us, that was a huge change because it forced us to change from a research project in my laboratory that might be useful to other people to a service infrastructure building a production resource that is broadly useful. I think we have seen, and I should also say that the driver for that, for the initial BRC program, actually predated the SARS pandemic. It grew out of the fact that in the anthrax attacks that in 2001, around the same time as the World Trade Center bombing, Congress mandated $1 billion to be spent on anthrax vaccines, which was a reasonable strategy and terrible tactics, leaving NIAID, including Tony Fauci, to try to figure how that money could be repurposed in sensible ways related to biodefense, one of which was to support the BRC program, which of course proved useful for many other outbreaks, including the cyclospora contamination of food and the SARS epidemic. Tony Fauci is certainly well known now. He was equally well known even before the BRC program for his pioneering role in addressing scientific research into the AIDS pandemic. And I think one of the things that we have seen, one of the huge successes of the SARS-CoV-2 pandemic, despite the appalling lack of public leadership in your country and my own, has been the sharing of data, right? The first sequences for SARS-CoV-2 were released in December. I know this very well because I teach an infectious disease and epidemiology class in the spring semester from January to May. And this year, starting from the first week of January, we were following the COVID pandemic. I'm sure that many of you have looked online at the popular epidemiology resources from Johns Hopkins and Our World in Data and IHME and elsewhere, and the various models. I think that is likely to have a strong impact. We have seen NIH launch many new initiatives for COVID studies, all of which have had a mandate for data sharing. Personally, I think the biggest challenge here will be everyone is now on board for data sharing in principle. Nobody knows what that actually means. So this is one of the things that the ClinEpi platform is designed to try to address, is to provide one vision for what it might mean to make data publicly accessible with an appropriate governance architecture that ensures that data is properly represented, discourages inadvertent misinterpretation of the data, and protects all applicable patient and study privacy laws, as well as the appropriate needs and for the bioinformaticists, epidemiologists, and others who have worked to make that data available. So I'm pretty optimistic about how these changes may transpire, and I am eager to try to evangelize for this vision of public data utility through things like the FluentEpi platform or what I think are demonstrable positive impact of the genomics platforms that we and other database providers have made available.
Thank you, David. Well, it seems that we don't have any more questions. And if that is the case, then I'd like to thank our invited speaker one more time for a wonderful presentation.
May I make one final comment, Joao?
Go ahead, David.
And it's for the other participants in the BMB meeting. So I have visited Brazil many times over the years. This is probably the longest time since that I haven't been to Brazil for 20 years. Of course, it's the longest time I haven't traveled for 20 years. And one of the reasons I've always enjoyed visiting Brazilian meetings, the Caxambu meetings of the Chagas Disease Society, the X meetings, and various other meetings in Brazil, is the fantastic quality of students and postdocs and the work that they do. But one of the things that's most frustrating about it has always been that for completely unjustifiable reasons, students and postdocs are often reluctant to ask questions for fear about in part for fear about their English language skills. But I am 100% certain that everyone on this call has English language skills that are sufficient for us to be able to communicate, certainly vastly better than my abysmal Portuguese. And so I'd like to encourage you to feel free to get in touch with. So this is what we miss by not meeting face to face, is that after a couple of beers or a caipirinha at the, what is it, Hotel Atlantic, was it? I don't remember, in Buzios. We would have plenty of opportunity to talk. It's a little bit harder by Zoom, but I encourage any of you to contact me by email if you have further questions, or particularly if you have tools that you think we might be able to help draw some attention to from the global infectious disease community.
Thank you, David. I'm sure that the many students that are listening in will take all your invitations and suggestions to heart, and hopefully by the time this pandemic ends, we will be able to meet again and have plenty of caipirinhas and fajitas and whatever else, and go on doing work on infectious diseases and databases. Okay, so as I was saying, it sounds like we don't have any more questions. So thank you, David, once again for having accepted our invitation. And Danielle, it's to you now.
Thank you, João, and thank you, David, for the excellent talk. We are finishing this session now. And see you tomorrow at the morning session. The morning session starts 9:30 a.m. So I would ask for authors to arrive 30 minutes before. So we have to test the share screen, okay? And so see you there. I'm stopping the